I've known him for many years. Initially, we served on the IBS board together many years ago, which was a lot of fun. And I've always been really impressed by his research because he's really made a huge effort to try to understand how past climate or past species distributions influence current patterns of species distribution or human behavior or what have you. And what's really special is that he actually goes after the data itself. So he goes after the fossils, or they do some ancient DNA, or some really exciting and unique and creative approaches to look at this question, which I hope he'll tell us about more today. And as a result of sort of his creative approach to this, he has numerous Nature and Science papers that sort of speak to the sort of cutting edge research that he does. And so we're really looking forward to hearing about this today. Thank you.
Wow, thanks, Catherine. I feel like I'm blushing now after your presentation. Well, it's a great honor to be here today. I know Catherine also for a long time, and other people in that room, like Nick. So it's great to be here today and maybe bring to you one of my research lines that basically relates to global genetic diversity. And I have been investing some time in the research line over the years. And of course, this is not only my merit, it's the merit of many of the people I work with. It's also a matter of chance by being part in the past of not only of the Center of Macroecology, but also of the Museum of Natural History, and now of a new institute called GLOBE, in which we'll have many other scientists around working on genomics, paleogenomics, and other aspects. So that has been also really important for me to be around these people to work on these kind of topics, right? And maybe we are not in my toolkit when I was starting as a researcher. So thanks, Rafael, thanks, Catherine, and your institute to have me here today. And I hope you find some of those things of interest, and then we'll have some feedback, and I'm much looking forward to know your opinion about some of the things we are doing here. So as Catherine says, I'm David. I'm part of the Center for Macroecology, Evolution and Climate. Yes, recently we are part of a new institute that is called the GLOBE Institute. For different reasons, we have ended up in the Faculty of Medicine and Health. Well, there are some university politics behind why we are there, but there are also some good aspects. The university leadership has the view of bringing biodiversity research into health and medicine aspects, and they are becoming more and more aware how medicine and health are basically depending upon biodiversity and other aspects. So that's our new adventure, and I hope it will go well. So, but let me start about this. I will present you today some results of my research and some ideas about genetic diversity and the global change. Coming from a global scale, that one is basically my background as macroecologist. So I guess no one in the room is going to find anything new in the next slides, at least, but as many of you know, their biodiversity is declining, and well, there are many different reasons, right? But if you think in the last 50,000 years, I love this plot or graphic from a paper in Science some years ago. Basically, we are taking over the planet, and we are, as a consequence of that, basically squeezing the biodiversity around us. And there are many, many footprints of our impact, both through climate change, but also land-use change. And as many of you know, the recent IPBES report has a really good work on summarizing what we are doing to nature around us. And there are really, really significant declines in many aspects of biodiversity, from population levels to ecosystem levels. As you can see here, in any case, there is not much mention to any genetic diversity aspect. The footprints are widely spread also across taxonomy groups. You can see here declines in invertebrates, for example. But if you move to other groups, we have a similar picture. We have more and more species coming to become more threatened. And that goes across the tree of life. And of course, it's a process that is not just something that has been going on over the last 10, 20 years or the last 30 years. What IUCN usually look at extinction since the last 500 years are quite significant across groups, and they are accelerating over the last century. And indeed, if we come back some thousands of years ago, we can see that the process is not even something that has started happening in the last 500 years. We have a large, mainly on mammals, a significant group, significant amount of mammals, basically, going extinct or declining largely in numbers because human and climate change impacts over the late Pleistocene and the Holocene. So when we compare all those trends over time, comes even more apparent that we are really living in times in which biodiversity is declining at a much faster speed. That's what we know, at least for basically the last 500,000 years, basically. And this kind of plot illustrates how the rate of extinction is accelerating over time. And even more worrying, right, is about also that predictions from models. And there are two people in that room that has been working a lot on predictions there for the future, like Catherine and Nick. They know really well that predictions, they don't look really good into the future. However, I think in many of these, much of our understanding in the decline of biodiversity globally is based at the species, is based at the level of species and ecosystem diversity. All of us, you know that biodiversity has three main levels: it's genetic diversity, it's species diversity, and it's ecosystem diversity. And somehow, some years ago, I started to appreciate that there was some gaps in the knowledge when it came to genetic diversity aspects and the decline of genetic diversity. And being genetic diversity a really important aspect of biodiversity, as it controls in many ways aspects of pathways of species going extinct. So it's quite interesting to see that in one hand we have a gap of knowledge at global scale on genetic diversity, but at the same time it has been recognized a really key role of genetic diversity to safeguard the functioning of the planet, right? And well, all of you might have seen this plot before. This comes from the planetary boundaries approach, in which genetic diversity basically has been always one of the main boundaries. But there is so much unknown. There are so many unknowns when it comes to estimate this boundary. What are the metrics to estimate this boundary? Collect information globally for many species. So we have this boundary in mind, but we are still not knowing that much. Similar for other major global initiatives to protect biodiversity, right? The CBD, Convention of Biological Diversity, has recognized for the last 30, 40 years the key role of genetic diversity. We have now the post-2020 framework for CBD. And again, many of us, we think that there is a missing opportunity there in what this post-2020 CBD framework means. I know CBDs start to realize about that and to move into a direction in which they are going to try to include more aspects of genetic diversity. So that's a bit where we are, right? So far, we have not been able to understand how genetic variation changes over space, continental, global scales. But at the same time, there are many good news coming. And it's because there is a new kind of discipline that is emerging these days. And I hope in the next 5, 10 years, we're going to have a much better understanding of how much genetic diversity is out there, what does it mean for conservation, and how we can use that to predict and anticipate impacts of global change. So there is a person in the audience today, I think, that has been the first author of a really cool paper. I think it's Deborah. I don't know if Deborah, I think I saw her around the speakers. Hi, Deborah, and many congrats for this fantastic review paper. So macrogenetics, it's quite a new discipline. And just a few weeks ago came what it's basically the first review on the discipline, right? So meaning that it's a really young discipline, but there is an enough body of work out there to start to realize about the potentials and the challenges. So macrogenetics, as Deborah and the co-authors did in this paper, it comes to be the study of large-scale patterns of genetic diversity and the processes shaping them. And when you can see here a really cool plot from the paper, it's a paper with amazing plots indeed. As you know, if you come back 30 years ago, when John Weiss and others were launching the phylogeographic research field, phylogeography has been always a discipline that has been mainly focusing in small systems, in one species or few species at local regional scales. And from there, we are moving now into a momentum in which over the last 30, 40 years, we have accumulated a really large number of genetic sequences. So this is making now feasible to really look at broader scales and to try to understand how genetic variation happens across space and maybe even in the future soon also across time. This has been, of course, something that didn't happen from nowhere. It's, of course, the work of many people and many different disciplines. And you can see here this really cool timeline in which we start in the 60s, basically, when scientists start to play with molecules. And from there, we have other disciplines contributing like GPS, satellites and remote sensing, of course, the ability to compile a lot of information and to share it like GenBank or GBIF, until basically moving what we have today. That is a new incipient discipline that is called macrogenetics. So this is just the intro to my talk, and in the coming half an hour, I'm going to start to explain how my research relates to these research fields, and starting by the first paper we published in my group on this topic that basically, and I appreciate a lot from co-authors, was mentioned here, and one of the first attempts, or the first attempt, maybe better or worse, to try to map genetic diversity for the planet. So let me start now at talking about global patterns of genetic diversity, and I'm gonna try to bring you back 10 years ago to a David that was 10 years younger and was start wondering about some of those questions. So around 2012, I was one day looking, I'm a geographer by training, so I love maps. That's what I would like to do, is to do maps. And I had in my desk maps of everything, maps of richness, maps of genetic diversity globally, maps of ecosystem diversity. And I was looking for a paper with a map of intraspecific genetic diversity, a global map for a big taxon, right? But I couldn't find any, and I got this idea, okay, maybe somehow we can do a map of intraspecific genetic diversity. So I applied for a grant, and the foundation somehow thought that was some kind of a good idea to do this, and we got funding to start working. So this was basically the main motivation of the grant. It's where is the intraspecific genetic diversity in our planet, to gain some kind of global view about it. And we thought that was really a need on that, right? I mean, as a macroecologist, I remember reading the papers from the 90s when people start to accumulate data to have the first global maps of species richness or the distribution of species. So I saw myself like, oh, wow, we need to start the process again, but now for genetic diversity. So thanks to the work of thousands of scientists across the planet that has been doing phylogeography or similar studies, we came into a point that was enough info with many constraints also to try to map this variation of genetic diversity. But we were facing really important challenges, what we call in the research group at the moment, the Gordian knot. We found that information on genetic diversity and responses are really fragmented across studies, that many of those studies were basically focusing on few species at local regional scales. And even more important, when you want to do a map, only a 5% of the sequences in GenBank at that moment have geographical coordinates. So the main challenge here was, how do we map with geographical coordinates? And we were able to do that. We were able to do that by building some algorithms that were able to go to GenBank, find information on the place that the specimen was taken from the field, and assign to that specific toponymic information geographical coordinate. So in doing that, we ended up with a map that got a lot of attention across academic disciplines. I'm really happy also that science thought that was worth enough to got the cover. So basically we were able to add geographical coordinates to close to 100,000 genetic sequences for mammals, amphibians, and I think there is other taxa in the paper. I remember now really well. And that made us really excited. It was also a kind of interesting and stressful process at the same time. I remember the day that the bioinformatician in my group called me, and after months of work, I didn't know he was able to design an algorithm that really would work and put coordinates to sequences in the map that would make sense. And I went to his office and I saw the first map. It's like, wow, yeah, this makes sense to me. So thanks to that effort, we were able to do the first global maps for mammals and amphibians, basically. And as you can see here, you have genetic diversity, which is a really simple estimate of genetic diversity. This is based on mitochondrial information. It's a really simple way to estimate nucleotide diversity, basically mutations per base pair, if you want to think about that, after controlling by species richness, right? Because that's a factor that can confound the pattern. So we start to find some of the first patterns there. They are not maybe many really obvious when you look at the grid cell level, but we start to show how the Amazonia seems to be a kind of area with higher levels of genetic diversity than may be expected by species richness. However, for me, the most fascinating map was the maps on the right, the maps of ignorance. And let me explain you what you see there. It's a 2-axis map. And in one map is the breadth of taxonomic information for a grid cell that goes to the bottom to the up. And the other, it's from left to right, is the amount, I think if I'm reading well, is the amount of genetic sequences we were able to compile. So what that means, if you look at the upper part on the right, you see Europe and some parts of North America in this kind of green color. And basically that means areas in which the taxonomic coverage and the amount of sequences are higher than in other places. And then we have regions like some parts of the tropics in which the taxonomic coverage and the amount of sequences that we were able to retrieve, it was really poor, meaning that there are areas in the world in which there is a lot of work to do. And a lot of work to do means also is coming back to the field. And well, there are some thoughts about that, how macrogenetics is gonna depend a lot of the ability of raising funds also to come back to the field. So we were trying to average these grid cell patterns to try to see if was an emerging pattern, right? By theory or given theories, in the relationship between richness, phylogenetic diversity, and other aspects of diversity, we were expecting to find latitudinal gradients. And we found that we see how higher latitudes, they host lower levels of genetic diversity after controlling by richness than the tropics, which is also fits with some theories coming from evolutionary biology. We had a second question in this paper that was, and there is some kind of human footprint that might be apparent. So we decided to use the approach of the anthropogenic biomes in the sense, okay, this defines some areas of the planet that has been modified in different ways, different intensity by humans. Here, the results are less clear in terms of, yeah, those areas that has been more heavily impacted host today lower levels of net diversity. We have some patterns there, areas that basically are cities or urban or more heavily impacted. They were hosting, in some cases, lower levels. So there is a kind of pattern there, but was not as clear maybe as we were expecting to find. So this was in 2016 when we published the first paper. So the next step for us is, okay, like in any macroecological study, first you find the pattern, then you try to explain the pattern. And we started to work on a new study that was more on what are the processes of space and time that has been shaping this global pattern of genetic diversity, in this case of mammals. So for this paper, we compile more sequences and we improve a bit the database. And we make basically a simple question, right? I mean, there are several candidates of hypotheses to explain this genetic diversity global pattern. Some of them relate to the evolutionary history. As you know, phylogenetic diversity and interspecific genetic diversity might be related, and there are theories about that, and we will talk a bit about these theories after. But there are also other possible factors. So one of my other research lines has been working with ancient DNA, past climate change, human impacts to understand biodiversity decline over the late Pleistocene. And we know that those drivers have been removing many populations, mainly mammals, and of course that is driving how much genetic diversity we have today. And then also we decide to add the idea of more recent human impacts, maybe over the last decades or century. So we lay out these potential drivers of the global pattern in this recent paper that was led by Spiros, was here. Indeed, Spiros did a PhD in Zurich at the Botanical Gardens with Comte, and then got a postdoc with us, and now he's doing really cool research in Frankfurt. And this is what we found basically. We had all this information spatially explicit about phylogenetic diversity, past climatic change over the Pleistocene, and recent human impacts. So we apply a couple of approaches, hierarchical partitioning and SAW, multimodal inference, to try to understand the role of these potential factors driving the global pattern. And We did the analysis at two different scales. In A is the grid cell level scale. B, it's a scale in biogeographic regions. We found that phylogenetic diversity maybe was the main driver. So processes driving the evolutionary history and diversity above the species was reflecting also the pattern of intraspecific genetic diversity quite well, similar for richness, but not as good. And we found also significant important effects of the variability of both rainfall and temperature. That comes for the grid cell level. If we analyze at the biographic region level, we are still having really important effects of evolutionary history, but the climatic aspects, they come of more importance. And well, that can happen, as you know, in biogeography. We have been studying a lot how different drivers change in relation to the scale at which you analyze them, right? So not surprising from that point, but in both cases, our expectations that evolutionary history and past climate will be at some extent behind the global pattern of intraspecific genetic diversity were confirmed here. So once we had this knowledge, we decided, okay, now we know what are some of the drivers, and even not perfectly, they are able to explain a significant part of the variation. Let's gonna use this to build predictive models so we can get estimates of genetic diversity in those regions of the world for which basically we don't have really good info. And what I mean really good info, it's places that you can see there with a white dot. This is places with really good info. We did a lot of sensitivity analysis to understand which places were. So the idea here, nothing novel, this macroecology has been doing this for the last 30 years, is to try to predict genetic diversity, which has been called also the Wallacean shortfall in areas for which we don't have knowledge. And this is the pattern that the model model came with, right? Tropical mountains are still coming up as the most highly diverse place in the world, which makes a lot of sense also. Then we'll have more tropical areas coming as basically major hotspots of genetic diversity. And then, of course, we see how that it's going down when we move to higher altitudes. So we did this for two different genes, because from the mitochondria, at least, there were the only two genes that with enough information to do this work. So as Deborah also mentioned in her recent research, I mean, it's important to have different, not only one gene, but have other kind of markers. And at that moment, that's what we had. And those are the patterns that they came from the model. So what our results were telling us? When we learned that multiple dimensions of normal diversity from the genetic intraspecific one to the phylogenetic diversity, they co-vary, they correlate across space. And this is not really far from some expectations in hypotheses like evolutionary speed hypothesis that even was framed for ectotherms, or for endotherms also, or the red queen hypothesis. So maybe not surprises from that point of view, but bueno, it's good to confirm that that's the case. We found some significant role of climate change, and this is easy to understand. Imagine a large mammal, which in the Pleistocene, late Pleistocene, was impacted by humans, but in a secondary way. But all, for example, the Arctic habitat in which this mammal was living, like the musk ox or other ones, basically collapse during the warming of the Holocene. So there are hundreds of populations that went extinct. That, of course, is going to reduce the amount of mitochondrial DNA that you can find. So also confirmation of a potential effect, also nothing new, I think, here. And we found no consistent effects of really recent human impacts, at least for the data that we have. There are some signals, but they are not really significant. So this makes me think that humans maybe will have a start recently to shape the pattern, but not at the level of evolutionary history and climate change over the last millions of years, right? But of course, that might mean that maybe it's not the case yet, but we are walking the way into that direction. And that basically brings me to, sorry, before I go into the future. For this paper that I mentioned here, we generate in Python an application in which you can find all the data and you can replicate all of our analysis, or including doing your own analysis. So, and it has a graphical interface to relate, as you can see here, gene, cytochrome B. You can play with different parameters, like how much of the taxonomic coverage you want to include, what is the minimum number of sequences, what is your response variable, what is your independent variable. So this is like a kind of way to provide readers with the ability to check all of our results, but also start to play maybe with other ways to explore our data. And I hope you find some of you useful somehow. So then we got into the next question, right? And this is again the classic macroecological path. You describe a pattern, then you come with some hypotheses that you test to try to understand the drivers of the pattern. And once you have some knowledge, you start to think, can I say something about the future? And this is, again, a work that we have been doing, and it has been published recently in Ecography a few months ago. So we start to think, okay, we have now maybe enough data, thanks to the model that we built in the previous paper, to have some estimates of genetic diversity across the planet. So how that pattern is exposed to climate change. And I would like here to repeat the word expose, right? I mean, there are different assessments, and the simple one, the most simple one you can do is just an assessment of exposure. Then you can move in sensitivity of a species. So, but that you need other kind of data. And then you can move into really hardcore quantitative extinction risk assessments. So this is just the first step in the process. So, okay, so from using the results coming from the previous paper, we have now this kind of model for intraspecific genetic diversity globally. And as you can see here, tropical ecosystems, of course, they host higher numbers of mutations per base pair, even after controlling, and you move to ecosystems in the north and you have lower levels. So using this info, and then using information on climate change and land use scenarios. And this was also one of reviewers really appreciate this part because we made the effort to take the scenarios that the new IPCC report just brought a few months ago in the publication of the new report. So it took us some time to, from scratch, being able to access to the scenarios and then to organize them. So we got up 18 or 19 models for future scenarios, both for climate change, but also for a new generation land use scenarios that has been harmonized, also for land use change. So imagine now we have in one hand a pattern of genetic diversity for different genes, and we have at the same time the new climate and land use scenarios for the planet. Basically, by linking these two aspects in a really simple analysis, but as I say, we prefer for this study to keep it simple, to talk about exposure, because we need a smaller number of assumptions. And also because that was the best we are able to do now. We came with a really simple kind of assessment, right? You cross these two maps and you can get this kind of maps. Again, you have there a two-dimensional axis map in which from the left to the right, you are changing genetic diversity from lower to higher levels. And then for the majority of the threads, you move from the bottom to the up, meaning more, less chain in the bottom, more chain in the upper part of that legend. So let's gonna focus on temperature change. So just to illustrate what these maps really are telling. If you see this really dark blue in the legend on the right, that means grid cells of the world that they are hosting high levels of genetic diversity, but suffering or that will suffer a warming more than 2 degrees, right? So those are areas highly exposed because you have high genetic diversity and high change in that parameter, in this case, temperature. If you see a blue color, it's an area in which you have high levels of genetic diversity, but the models don't predict a big change on temperature, right? So they are less exposed. Some areas, for example, in Southeast Asia, they are, from that point of view, in temperature terms, highly rich in genetic diversity, but less exposed to temperature change. Of course, global change is a matter of different drivers. And then if you move into the next thread, that is precipitation change, you see other picture. You can go to the same areas of South Asia, and of course, maybe the temperature will not change much, but they are gonna suffer big changes in rainfall. So that, of course, creates a higher level of exposure. And we did that for basically different climatic parameters and different human parameters. We were really tempted to unify all those things in one single map, but well, we thought that might be more confusing than helping, and we decided finally maybe not to include that. So this is up to our knowledge, maybe the first global assessment of global change on genetic diversity. Is only for mammals and is based on model data for genetic diversity, but it's still relevant. And if you go to the manuscript in ecography, you will find many more details and quite a lot of sensitivity analysis about what we found. But something that make us really excited is that really the message went through. And we got a lot of attention about this paper just by saying, yeah, there is a significant exposure to both climate and land use change. So a few days later, after the publication of the paper, we were contacted by the World Conservation Monitoring Center in Cambridge, which is part of the United Nations Environmental Program. And they came to us basically with the request that was as follows: like, we are soon having the meetings of the parties of the CBD. We have a lot of indicators and assessments for other levels of biodiversity, species, phylogenetic, of ecosystem diversity. So far, we don't have any global assessment. We understand your assessment is still quite simple, but we don't have any other thing. So for us, it will be really important if you would allow us to bring your results into a new indicators website that will be used for the discussion of the parties and also for more widely used across policy makers and managers and other colleagues, right? So this is, of course, exciting for us because it's our research, but I think it's exciting also for the field of macrogenetics. They are really in a pressure to have these indicators. Here, of course, there is a trade-off, right? They are telling us basically that even your indicators and data is still really primitive. It's better than nothing. Because they are asking me now, okay, now can you replicate that for the last 1,000 years and you can have sequences over time and can you go into full genomes? It's like, yeah, we can do all those things, but it's going to take between 5 and 10 years many colleagues around the world, yes, will be, right? But so they understand the limitations. And I think for macrogenetics, my view is like, meanwhile, we move into more complex data and assessments, we really need to try to make an effort to populate CBD and other major initiatives with some kind of data. And that's something that I think is gonna open a lot of possibilities also for funding in the coming future in the field of macrogenetics. So what is the road ahead, right? And with this, I will finish my presentation. So of course, all of us, we are moving from this mitochondrial to whole genome data. It's not an easy task. There are more and more whole genome data out there, but still much less than the mitochondrial data, because mitochondrial data has been there for 30 years, right? So we're still really far using whole genome data to have the same taxonomic and spatial coverage. It will take, as I say, 5-10 years. As you know, what means mitochondrial diversity for conservation is a topic of massive debate. When you go to talk to people working on population genetics, they don't agree. They didn't agree for 30 years. We know that the mitochondrial, it's in theory neutral, but recently we start to know that the mitochondria also plays a role in proteins and in the adaptation to some heat events, for example, so it's not fully neutral. But I think we're going to see more and more, and it's important to move into functional genomics, patterns of adaptive genetic variation. And there is some preliminary work on that, maybe not for many species, but that's where we are moving. I don't have time today, but my grant, I'm still calling my new grant, even started a few years ago, because with Corona, we couldn't do anything. So it's still looking like new. But for example, I have a new grant that what it's doing is sequencing at the whole genome level around 500 birds specimens across the planet. So for 100 species. So for each species, we have 5 populations. That will help us to answer questions in relation to paleo demography, genomic functional adaptations of local populations. The project was heavily relying on museum collections. And with COVID, all the museums in the world has been basically closed down. So mainly the museums in US. So bueno, now they start to be back on work, and we are starting to get in Copenhagen the samples to do the sequencing. This is an initiative in which the other co-PI is the director of the BGI Center in China, Wu Yisang. Well, they have a really massive infrastructure, and it happens that also is in the first floor of my building. So that's what I'm saying, that this is not just me. It's also lucky enough to be in a place in which I have people around that also have the means and the knowledge. And of course, maybe the third axis is to move from a static picture in time to a temporal picture in time. And there's when we have the ancient DNA and even things that are even really new with paleoproteomics, where you can come back maybe 1 million years back on time. Say, this will be really important, right? We need also some temporal perspective. When I have been in some workshops, or so involved in some workshops, even if I was not able to participate properly, I remember GeoBond with Enrique Pereira organized around these ideas a couple of workshops and on biodiversity, how they call it, EBBS, essential biodiversity variables. They want to have a number of metrics for genetic diversity, and of course, they also like to have temporal trends. So, well, there are some ways to maybe have temporal trends, now thinking on hundreds, thousands of years. but there is a lot of work to do, right? And you can see here this road ahead. It's really straight. Maybe I didn't pick up the best picture. Maybe that road should be more like a curvy, I might say, right? There's an exciting time ahead, but a lot of challenges. And that's also why I was really excited, Deborah, about your review. I guess now I can say that I was one of the reviewers of your paper, and I thought, wow, yeah, this really is giving us now a broader view of what is ahead. It's really exciting, but will not be easy. And bueno, there are a lot of people I should be thanking, and I wouldn't have pictures enough. So yes, a really small core group of people that has been part of these 3 papers I have presented over the years. And of course, to the funding bodies, the Danish Research Council and some private foundations in Denmark. So I think that's all I have for you today. Many thanks again for letting me present my research. And I happily talk to you now about any comment or question that you might have.